Hello everyone, welcome to the session. This is Om Prakash Shetty, Advisory Director of AgriExplorer. AgriExplorer is an initiative for students where different activities are performed for students of agriculture, biotechnology, and a related field. AgriExplorer provides internship opportunity to these students. Today's session is a part of internship. Richa Das is our today's speaker and will talk on the topic fermentation equipments and its uses. Without taking much time, I welcome Richa to have your presentation. If you have questions, then please drop it in the chat box. Richa, you can start now.
Yes, sir. Uh, Thank you, sir, for the introduction. This is Richa Das. I'm studying in Parul University, Vadodara, Gujarat, and I'm doing bachelor's in biotechnology. First of all, I would like to thank AgriExplorer for giving me this opportunity. I love researching and teaching, and I think that this is the platform that is providing me both the things simultaneously. So, thank you to the team, to the entire team of AgriExplorer. The topic of discussion that I'm going to present today is fermentation equipment and its use. So before I move on with my slides, let me tell you all that why I have chosen this topic. We human beings, we all always we demand for the best things. We always want to have access to the best things, whether it is food or whether it is lifestyle. So fermentation is the process where microorganisms will be used to manufacture a variety of products, and these products are going to be high in quality. Are going to be their nutritional quality will also be good. So let's suppose that if today I have discovered a microorganism that is giving me a product. And let's suppose that this product is by far the best product apart from the previous ones. So my aim should be that how am I going to isolate the organism? How am I going to prepare pure cultures of it? And most important is that how am I going to use this microorganism to get my desired product? So fermentation. So that is why this topic I have taken, because it's very useful and it's very important to know that why the fermentation processes are carried out and how they will be beneficial to us. However, in this today's session we will be discussing about that what is fermentation, what is a fermented, what are the various steps of fermentation, what are the parameters of an ideal fermenter, and also various parts of the fermenter. So moving forward, I have already discussed the contents just now. Okay, so moving with my first slide, what is fermentation? Fermentation, as I said just now, it is a process where microorganisms are used to give out a product. So basically, it is said that fermentation is a metabolic process. First, let me stress upon this term that what is metabolic processes. Let it be any living organism, whether it is human beings, animals, or living organisms. Metabolic processes are very important to sustain or to continue their life. So we can say that metabolic processes are the biochemical processes in which whatever the food, whatever the nutrients we are ingesting, not only we, even whatever the microorganisms are ingesting, they will be converted, they will be broken down, and they will be converted into some desired products. So this the same thing is, so this is called metabolic processes, because after the things will be broken down, the food items will be broken down, that is going to give energy which will be utilized by the organisms, by the cells of the organisms, and also it is going to give out a product. So this metabolic process is exploited in fermentation. That is why we say that fermentation is a metabolic process where microorganisms are going to ingest, are going to consume food items, which is probably we can say sugar, and these sugars will be converted into various products, like it will be converted into acids, gases, or alcohols, and other products. Also antibiotics can are also made in the same principle. Moving to the next point, as I said that fermentation requires microorganisms. Now fermentation is a process that is always done at a commercial rate. For example, that we can say Amul Industries or Coca-Cola Industries are there. Whatever the product they are producing, they are not producing it for like only for a particular number of people. They are producing it for the entire nation. So whenever anything is done for the entire nation, that means whenever anything is produced commercially, to do the process of fermentation, the microorganism should also be required in lots of amounts, because then only my desired product will be produced in high quantities. So the next point says that it's that fermentation is the process where bulk of microorganisms are grown. That means millions and billions of microorganisms will be utilized to give out our product. And the next point says that zymology is the science of fermentation. Okay, this screen is showing various products like curd, cheese, alcoholic beverages, vinegar, etc. And all these products are the product of fermentation. It is the fermentation due to which we are getting these products. So the products are not only limited to what is present in this screen, but there are some more products which I have not presented here. That is antibiotics. As I said, antibiotics is also one of the uh, fermented products. So the first image we can see is of the curd, where lactobacillus organism, the microorganism that is used to do the fermentation of curd, that is lactobacillus. Then for cheese, initially we use as for preparation of curd, preparation for cheese, curd is used. So initially lactobacillus will be used, but after that many other microorganisms can also be used there. Then for alcoholic beverages, we make use of an yeast, that is Saccharomyces cerevisiae. And vinegar, the next picture is of the vinegar. Vinegar is actually a product of alcohol only. So these are the images that show us the various products of fermentation. Now we will see what are the various steps to carry out fermentation. Okay, to the first point. So fermentation, as I said, it is a process where microorganisms will be used, and we have to make the microorganisms to feed upon some food item, which is probably sugar. So our first step is that we have to prepare this food item. We have to formulate this food item, and this food item is called as the production media. So our first step is that we have to prepare an appropriate production media, because it is this production media that is going to contain sugars, macronutrients, micronutrients, vitamins, etc., upon which the organism will feed, and then our XYZ product will be given out. The next step is saying that once we have prepared our desired product, the production media, once we have prepared the media, the food item upon which the microorganism is going to feed upon, our next step is to sterilize the production media, because while the media was getting prepared, chances were there that some contamination might have occurred, and we don't want such contamination to occur when we are manufacturing products. That is why the production media is to be sterilized. The fermenter is also to be sterilized. So let me tell that what is a fermenter. We are going to discuss about fermenters in our next slides also, but just to give an idea, that fermenters are actually very large vessels which are either made up of glass or maybe stainless steel, inside which the whole process of fermentation takes place. So we have to sterilize the media, the fermenter, and as well as the ancillary equipment. So here we have a new term, ancillary equipment. This term would mean that whatever the pipings, whatever the fittings are there in the fermenter, all those pipes, all those fittings should also be sterilized. So those pipings, those fittings, the screws and everything, they come under the ancillary equipment. Okay, the next point says, now once we are done with the preparation of our media, sterilization of the media, now the next point is growing our microorganisms in millions and billions. Because just now I said that fermentation is a process, it is a commercial process. To do that, we will require microorganisms in millions. So our next step is that whatever the pure culture we have isolated from a mixture of microorganisms, now we have to multiply. We have to allow these microorganisms to grow and divide, so that we will get sufficient amount of microorganisms. Now once you get sufficient amount of microorganisms, like millions and billions of microorganisms, then only the actual process of fermentation will begin. Okay, the next step is, now once, as I said, you have sufficient amount of microorganisms, the next step is starting the process of fermentation. Now we very well know that for any living organisms to live, either it is animals, plants, human beings, or microorganisms, we all require some optimum conditions. We require optimum temperature, pH, nutrients, and as well as proper amount of oxygen as well. So our next step is that the growth of the microorganisms will take place in the fermenter only when all these optimum conditions will be provided. Moving to the next step says, now once the product has been made completely, okay, now let me tell you that when the process of fermentation takes place, it is not a 1 day process. It can be a 1 day process, but it can be a 5 day process also. So once after 1 day, 2 days, or 5 days, your process is completely done. The next step is to extract out this product and to purify the product, because inside the fermenter you not only have your product but you also have your byproduct. So it's very important to separate my product from the byproduct, because byproduct is something which is an effluent. It is actually a waste thing. We don't want that. We want the product, so that needs to be extracted as well as purified. And the last step is that whatever the byproduct, which is termed as effluents, we have got these byproducts, these effluents need to be treated and then they will be disposed of. But let me tell you that nowadays effluents are not disposed, because because we already know that how we are polluting the environment. That is why nowadays the effluents, whatever the byproduct or waste substances is getting released, that is given to some other industry. So what is the so basically uh, the any byproduct which is a waste substance for us can be useful for some other industry. So these were the steps of fermentation. So in this picture we see in this picture the whole process of fermentation is described. So we see that the large body that we see here, this is the actual fermenter where the process of fermentation takes place. And this blue color which we see, this is called as the downstreaming process. Just now I said that the product needs to be extracted, it needs to be purified. So all those processes are downstreaming processes. So starting from the first image, that is stock culture. So let me tell you that what is stock culture. As I said that microorganisms will be required in the process of fermentation. That means I have to isolate my organism, my pure form of organism from the mixtures. So and then have to cultivate as well that pure form. So that pure form of organisms which is containing only my desired microorganism is called as the stock culture. Now once you grow the microorganisms in sufficient amounts, then we put it into something called seed fermenter. So what is a seed fermenter is that before we actually start the process of fermentation, we have to first produce our product in smaller amounts to see that whether my desired product is getting produced or not. Because sometimes it happens that I have isolated my organism, but instead of product X, product Y is getting synthesized. My desired product is X, but instead of X, Y is getting synthesized. So that is a failure, and we don't want such scenarios, because if directly we use the large fermenter, then we are not only wasting time but we are also wasting a lot of money to give out a wrong product. That is why after you have isolated your pure culture, the next thing we will do is producing our product in smaller amounts. And then when we observe that our desired product is getting produced, then only this large fermenter, as we can see, this large fermenter will be used then only. Now after that the production is going to take place. Then the culture fluid will be removed. Culture fluid is here the production media which I spoke about in the first point, which is the food item for the microorganism. So culture fluid needs to be removed. Then from this culture fluid we have to separate out the cells in the sense cells of the microorganisms. The microorganisms needs to be extracted out, and then the downstreaming process will start, where we are going to separate our product from the byproduct. The byproduct will go for the effluent treatment. Then it will either be disposed or it will be given to some other industry or country. Then the product will be purified, and finally my desired product will be packaged, and then it will go to the markets. So this is the whole process. Okay, now we will see that what is actually a fermenter. So just now in the previous slide I said that fermenter is that large vessel or that large apparatus inside which the process of fermentation takes place. So fermenter sometimes they are also called as bioreactor. Actually, there is a minor difference between fermented and bioreactor. The minor difference is that the term fermented is used when we want to grow microorganisms, and the term bioreactor is used when we want to grow plant cells or animal cells. Otherwise, so there is just this minor difference. Otherwise, the way they operate and the design of both these fermenter as well as bioreactor is the same. Even the way they operate, the operation of fermenter as well as bioreactor is the same. The only the minor difference is that fermented term is used for microorganisms when we want to grow microorganisms, and bioreactor is for plant cells or animal cells. In many books we will find that fermenter and bioreactor are used synonymously. Okay, now let us see that why we require fermenter. So the uh, so as I said, that we are using microorganisms in the process of fermented, and we have to provide all the optimum conditions to the microorganism, because then only it will be able to synthesize our desired product. So those all those optimum environmental conditions can be maintained inside this apparatus, that is your fermenter. That is why that is why the fermenters are used, because inside this fermenter only all the optimum conditions will be provided to the microorganism, and then only the microorganism will be giving out various products like enzymes, antibiotics, organic acids, alcoholic beverages, and etc. Okay, so these points I have already discussed right just now, that they are used synonymously. There is a minor difference between them. What is the minor difference? That also I have said. And as I said, that the operational parameters, the way they operate and the way they are designed, both are same. Now we will see what are the parameters of an ideal fermenter. Let us suppose that I want to set up an industry, or you all want to set up an industry, and you all are going to the market to buy a fermenter. So there will be thousands of fermenter. How are we going to decide? How are you going to decide that which is an ideal fermenter? So these are the parameters on the basis of which you are going to decide ki, yes, this is my ideal fermenter where I want to carry out the process of fermentation. So moving to the first point. Okay, my ideal fermenter should be such that I can grow any type of organisms inside the fermenter, and I can make any type of product inside that fermenter. For example, that if today I have used a fermenter to make curd, let's suppose, then tomorrow I should be able to use the same fermenter to make alcoholic beverages, or let's suppose teas. So this is only going to be my ideal fermenter. So my ideal fermenter is such that I can grow any type of microorganism. Let us suppose that today, if I am growing lactobacillus, then tomorrow I should be able to grow Saccharomyces cerevisiae in the same fermenter. So that is an ideal fermenter. Next point is that an ideal fermenter should be such that it should not allow any unwanted microorganisms inside the fermenter while it is getting operated, while it is functioning. Because if any minor contamination also takes place, we have to discard everything. So that is going to waste a lot of time and money as well, as I already said. The next point is temperature maintenance. So maintaining temperature is very important. My ideal fermenter should be such that it can maintain any temperature, whether it is 26 degrees Celsius or whether even if it is 55, then also my fermenter should be able to maintain that these temperatures. Okay, the next moving to the next point. Okay, my ideal fermenter should be such that it should provide both aeration and agitation to the microorganisms, because then only they will be able to synthesize the product. Now, first, let me stress upon these terms, that what is aeration and what is agitation. The term aeration means providing sufficient amount of oxygen. If you are using aerobic microorganisms, then it's compulsory to provide enough amount of oxygen to them. So that is called aeration. And agitation would mean the phenomena of mixing. Let us understand this term like this, that in our homes we use mixer grinders. So what is happening in the mixer grinder is, when we switch it on, whatever the contents you have added in your blender, uh, it is going to agitate, that is, it is going to mix. So Agitation, the meaning of agitation is mixing the contents, or agitation of contents. So I think that these two terms are clear, that what is aeration and agitation. So coming to the point, coming to the fourth point now, that in the process, in my ideal fermenter should be such that it should be able to provide both aeration and agitation to the microorganisms, because then only my desired product will be synthesized. And while aeration and agitation is being provided, the microorganisms should not get damaged, because if they get killed, then your product will not be synthesized. Now, how aeration and agitation are used in fermentation, that we will discuss in the next slides, when we will be discussing the various parts of the fermenter. The next point is maintenance of pH. So just like temperature, appropriate pH is also to be maintained, whether it is acidic or basic. Then the next point says that my ideal fermenter should be such that whatever the levels of oxygen are there inside the fermenter, I should be able to monitor that, because initially whatever oxygen we will be inoculating, we will be putting inside the fermenter, that is not going to last till the whole process of fermentation. Repeatedly I have to supply oxygen. So I have to, so my ideal fermenter should be such that it should allow me to monitor that what are the levels of the what are the levels of the oxygen, because according to that only I will be able to inoculate oxygen. Next point is, uh, my ideal fermenter should be such that it should allow me various access points, various feeding points through which I can inoculate my nutrients or the production media. So let me tell you here that in the fermenter, when we are inoculating our production media, we are not going to open the main lid of the fermenter, because once you open the main lid which is present at the top, then unwanted microorganisms can also enter. So how do we actually enter the nutrients and our production media is, there are pipes present. It is through these pipes how my ideal fermenter should be. Okay. Next point is power consumption. So yes, in the process of fermentation, power will be used. So my ideal fermenter should be such that it will consume less power. Next point is that my ideal fermenter should be such that it should use less amount of labors, because when to handle a single fermenter, let's suppose 10 people are there, so chances of contamination, risks of contamination will be high. So that is why I have to buy such a fermenter that will be fully automatic, that is going to operate itself. So there are various sensors fitted into the fermenter nowadays. Sensors are there for temperature sensors, pH sensors are there. So and oxygen sensors are also there. So nowadays whatever the fermenters are available in the market, manual power is not at all required. So that is how my ideal fermenter should be. Okay. The last point is that when the body of the fermenter is constructed, as I said, that glass or stainless steel will be used. Now if whether it is glass or whether it is stainless steel, a single sheet cannot be used. Let's suppose if we talk about stainless steel, a single sheet of stainless steel cannot be used. We have to use multiple sheets of steel, for example, two or more than two sheets also. Now how are we supposed to stick these two sheets? So there are two options for us. Either we can use screws to stick these two sheets while we are preparing, while we are making the body of the fermenter, or we can do welding. So the term flange points here, we can see flange points means screws, which I said right now, and the next option is welding. So I think we all might know that what is welding, but still I'll give a gist of what is welding. Welding is a process where the metal will be heated, and while we are heating that metal, they are going to start melting, and by melting we are going to stick those different parts of the metal. So that is called welding. So now coming to the point, we have two options to stick our metal sheets. Either we can use screws or we can use welding, but what is mostly preferred is welding. Why screws are not preferred is because when screws will be used, the screws will be large in size. Chances will be there that the production media will start accumulating around these flange points, around these screws, or maybe your microorganisms will also start accumulating in upon these screws. So we don't want such scenarios. We want that the internal walls of the fermenter should be as smooth as possible. So to make the surfaces very smooth, welding is preferred. Mostly screws are not preferred. And the last point for the parameter is that whatever the material has been used to construct the fermenter, that material should be inert. I think we know what is inert. Inert means that when any material is itself not participating in the reaction, because when the process of fermentation is taking place, inside the fermentation many metabolic processes, enzymatic processes, chemical processes will take place, and we don't want the walls of the fermenter to start reacting in those reactions. Otherwise your product will be a poisonous and a deleterious product. We don't want those effects. That is why the fermenter should be made up of such a material that is going to be inert. Okay, so this is how the basic design of the fermenter looks like. Generally they are cylindrical in structures, and there are two materials, glass and stainless steel, by which they will be made up of. So we see there are different labelings here. However, we are going to discuss these labelings and these various parts in our next slides. But as you can see that here there are some pipe like structures at the top, and also here the top is the aseptic inoculation pipe, and here also we have the sampling point. So these are the ancillary equipments which I told in the previous slide, and these are the points through which I am going to inoculate my production media. I'm not going to open the top lid because that will cause contamination. Okay, so these are the we will see some different types of fermenter. This is a lab scale fermenter, which I think we all might have seen in our universities. Lab scale fermenters are actually made up of glass only because they are very small. We can carry them from one lab to another lab also. Then we have the pilot scale fermenter, which is bigger than the lab scale fermenter, but it is smaller than the your main production fermenter, and they are mostly made up of stainless steel. And then these are the main fermenters about who which I was talking in the about the ideal fermenter. So these are the production tanks or the main fermenter where the actual process, commercial process of fermentation takes place, and they are actually very large. They might be 20 to 30 feet tall, or they may be more as well. Okay, now we'll be seeing various different parts of the fermenter. So there is something called headspace volume. Any fermenter is divided into two parts. One is called as the working volume and the other is called the headspace volume. Let me show the diagram here. So we can see that this is the fermenter, this is the body of the fermenter, and each and every fermenter is divided into two parts. That is a headspace volume, which is at the top, and the working volume. Working volume is that space in which you are going to have your production media, microorganisms, as well as air bubbles, whereas this headspace volume is the one which is going to be empty. Okay, so uh, the proportion of this working volume is said that it occupies 70 to 80%, as we can clearly see in the diagram that most of the space occupied in the fermenter is by the working volume. So working volume occupies 70 to 80% of the space, and the remaining 30 to 20% of the space is occupied by this headspace, as we can see above. Okay, now let me tell you that why. So first of all, let me show you another diagram that will give a clear picture of what is the headspace volume. So this is a fermenter, a glass fermenter. It is made up of glass, and we can see that this top portion, which is empty, this is the headspace volume, and this portion, which is having your production media as well as the microorganisms, this portion is the working space. So I think that it is clear that what is the headspace. So this part is the headspace and this is the working space. Now let me tell you all that why headspace volume is important. Let us understand it like this, that when in our homes we make use of a mixer, we keep a lid at the top of the blender. Why? Because when you will switch it on, the contents will start jumping. So that's why we keep a lid at the top, so that the contents will not come outside or they will not jump. So what happens in the process of fermentation is that when mixing is taking place, when agitation is taking place, this media, as you can see, the media that is present in this working space, this media will jump. And chances, and if I fill this media up to this point, up to the if I fill the my production media till the whole brim of the fermenter, that means till here, let's suppose, then when agitation will take place, this media will jump and it will stick upon the inner top surface of the fermenter, that it will start sticking here. And this is actually undesirable, because when your production media will start sticking at the top, it will start dripping, and maybe the microorganisms will start accumulating at the top lid also. You will not get desired product, or maybe you will get, sometimes you can get a wrong product also. So that is why headspace volume is maintained, so that so that's why this headspace volume is maintained, so that when the process is going on and when your production media is dumping, it should not stick at the inner top surface of the fermenter. That is why it is done. Okay. And so that is the first thing why we require headspace volume. The second reason why we require headspace volume is that, as we can see in this diagram, in the headspace volume there are some foam like structures. So these are, so foam production actually is very common in the process of fermentation, whether you are manufacturing curd, um, antibiotics, or maybe alcoholic beverages. Foam will be produced, but the amount of foam produced will vary from product to product. For example, that foam formation in curd will be minimum, but foam formation is very high when you are manufacturing beer, when you are manufacturing wine or vodka or any alcoholic beverages. Then the formation of foam is actually very excessive. So that is why, depending upon that how much foam is forming by the product, like that we are going to adjust the headspace volume and the working volume. For example, that if I am manufacturing curd, then my working space will be 70% and my headspace volume will be 30%. But let's suppose if I am making alcoholic beverages, then my working volume should be 30% and my headspace volume should be 70%. So headspace, the proportion of headspace volume and working volume depends upon what type of product we are synthesizing. And if your product produces lots of foam, then it is advised that we have to keep the proportion of the headspace volume should be more than the working volume. So all these points I have discussed just now. Okay. The next part of the fermenter is the foam control system. Foam control system would mean that, as I said just now, that any product you are manufacturing, foam will be formed, but we have to prevent the formation of foam, because what happens is that when excessive foam is forming, it will start blocking the air exits. Now, what are air exits? As I said, that oxygen is supplied inside the fermenter, right? And so when there is a accumulation of large amount of air, if large amount of air is getting built up in the fermenter and that air is not able to escape outside, a pressure will build up, an unwanted pressure will build up inside the fermenter. And because of this unwanted pressure, what will happen? It can lead to the damage of the microorganisms, it can kill the microorganisms, it can loss of production media can also happen. Most importantly, sometimes because of such pressure buildup, fermenters get blast, and blasting of fermenters is actually very injurious uh, to all those people who will be present in or outside that facility. So that is why we have to minimize the foam, because if the foam keeps on building, the air exits will be blocked, air will not be able to move outside, and pressure will start building up. So that is why it's very important to minimize the foam. So how can we minimize foam? Is by using silicone or vegetable oils, we can actually minimize the concentration of foam. But silicone can be harmful for the product as well as for the microorganisms. So basically, we generally choose vegetable oils upon silicone. So silicone and vegetable oils are called as anti-foaming agents, which will be utilized to minimize the formation of foam inside the fermenter. Okay. Okay. So the first 3 points I think I have said just now. Yeah. Okay, the next point, that is the fourth point, says that we have to, it's very important to use antifoaming agents. But if you keep on using excessive antifoaming agents, then proper oxygen transfer will not take place within the microorganisms. And we don't want this. We want that there has to be a proper diffusion, there has to be a proper transfer of oxygen. So that is why excessive foaming should also not be used. Okay. Now, as I said that nowadays we use fermenters which are fully automatic. They are fitted with sensors and everything. So here also there are some foaming probes or sensors. So here we can see the word probe. Probe actually means sensor. The other term for probe is sensor. So foaming sensors are also kept. So let me explain that what is the significance of these foam sensors is. There are two sensors. One sensor will be kept inside the fermenter at the bottom, inside the liquid media, that is here. First of all, that both the sensors are going to be fitted inside the fermenter only. So the first, so one sensor will be situated here, and the other sensor will be fitted here at the inner top surface of the fermenter. Now what will happen is, when the foam production will start, if this foam production hits the this sensor, then the sensor will come to know that excessive foaming has been formed. Then what will happen, there are nozzles which are situated outside the fermenter. Those nozzles will open, those taps will open, and there are actually pipes also situated outside the fermenter. So through these pipes, automatically the anti-foaming agent, that is your vegetable oils or maybe silicone, will enter inside the production tank. So this is how sensors are used to automatically add the anti-foaming agent. So we again, manual power is not required here. Okay. The next part is the temperature control system. So as I said that inside the fermenter, microorganisms are growing, and it can be a one day process. I mean, the manufacturing of the product, that can take 1 days, 2 days, it can take 5 days also. So what happens is, the temperature is never consistent. The temperature can keep on decreasing, it can keep on increasing. So when the temperature is increasing, that means when the temperature is out of its optimum, that means, let's suppose that optimum temperature should be 26, but now the temperature is 55. So we have to bring down the temperature into its ideal, into its optimum or ideal condition. So for that, there are cooling jackets or water jackets which are kept outside the walls of the fermenter. Again, there are sensors present. So when the sensor will come to know that temperature is increasing inside the fermenter, nozzles will open, taps will open, and through the pipes, water will be inoculated in the cooling jackets. Let me show the diagram of the cooling jacket here. Yeah. So as we can see, this yellow color or orange color portion, these are the cooling jackets which are situated, which are kept outside the walls of the fermenter. So when the sensor will come to know that temperature has been increased rapidly, the nozzles will open, the taps will open, and water will be coming through the pipes, and then they will be entered into these cooling jackets. And then the temperature of the fermenter will be brought down to its optimum or ideal temperature. That is how, so that is the temperature control system. The next part is the agitator or the impeller. Okay, so let us start like this. That again, the same example. In our homes, we have used mixers. So inside those mixer, we have seen there are some blades present. So what a blade or fan-like structures are they? So what is the significance of those blades or fans? Whatever the content we will be inoculating inside the fermenter, I mean inside the mixer, these blades are going to break those contents. For example, if I want to grind spices, spices will be entered, will be put inside the mixer. I will switch it on, and these blades or these fan like structures are going to crush the spices, and it is going to create a homogenized mixture of all the spices. I think we know what is a homogenized mixture. Homogenized mixture would mean that where anything is mixed completely such that we cannot separate them, or we cannot find their identity. So that is called a homogenized mixture. So The same thing is used in the fermenter as well. Even the fermenters are fitted with these fan like structures or blade like structures, so they are called as agitators, or we can say impellers as well. Now what happens, the same function is being done. Whatever the production media we enter, we inoculate inside the fermenter tank, we want that there should be a proper homogenized mixture. So to maintain a proper homogenized mixture, impellers or agitators are fitted inside the fermenter. Um, then for proper oxygen transfer, we want that there should be a proper transfer of oxygen, or proper transfer, proper diffusion of oxygen part. So for that also impellers will be utilized. Okay, so these are the types of agitators or impellers, that is disk turbine, van disks, open turbines of variable pitch, and propeller. So let us see the diagram here. So the right image is how an actual fermenter looks like, and these are all the blades of the impeller, and these are the various types of the impeller. The first one is the disk turbine, then we have the van disks, we have the open turbine, and then we have the propeller. Out of all these four, it is the first one, that is disk turbine, that is considered to be the most suitable one. Okay, so now there are two important functions of this impeller. The first function is that whatever oxygen, whatever air we are inoculating, we are putting inside the fermenter, these oxygen bubbles will be very large. And because the size of these bubbles, size of these oxygen bubbles will be very large, their diffusion, their path of diffusion will be hindered. They will not be able to transfer among the organisms properly. So the job of this impeller is to break down these large bubbles, large air bubbles, and to convert them to smaller ones, so that they will have a greater surface area to transfer among the microorganisms. That is the first function. Second function, as I said, that we have to prepare a proper homogenized mixture. Whatever nutrients or sugars you have provided in the production media, we want that it should be thoroughly mixed with each other. Now why we want this? Because we have to make sure that each and every microorganism gets equal amount of all the nutrients. And that is why impellers or agitators are used, because they are going to mix the content thoroughly and properly, such that each and every microorganism will be receiving equal amounts of those nutrients. Okay, the next part of the fermenter we have is the baffles. So let me show the diagram here. So these are the baffles here. We can see baffles are situated, they are kept in at the inner walls of the fermenter, as we can see here. These are the baffles. This is the internal picture of the fermenter, and here also we can clearly see the impeller, the baffles here. So these are the baffles, which are metal stripes. Here also we have one more baffle. In this picture we can also see there is an impeller here, agitator. This agitator is attached to a shaft, and this shaft is situated to a motor like this. So when we'll switch on the motor, the shaft is going to rotate, the impeller will also rotate, and then the process of agitation will start. So this is the impeller, this is the shaft, and these are the baffles, which are the metal stripes. Okay, now these baffles should be 1/10 of the fermenter. Why they have to be 1/10 of the fermenter? Because if the width of these baffles are too large, then these baffles and the blades of these impeller will collide with each other, and they will hinder each other's activity. So that is undesirable, we don't want that. That is why if you see that these baffles are 1/10 of the fermenter, so that these baffles will not collide with these the blades of the impeller. Okay, now let me tell you that what is the function of the baffles, because actually baffles are also used for mixing the contents. But I think this question might be that if already impellers are kept inside the fermenter for mixing the contents, then why do we require baffles? The answer to this question is that this impeller is occupying only a certain portion of the fermenter. So whatever the contents are there within the reach of these impeller, only those contents will be mixed. Okay, but what about the contents, what about the production media that is present at this part or at this part? Because if you will see the diagram, as I already showed earlier, the diagram of the large production fermenters, they have very big diameters as well, and you cannot use large impeller, because then these blades of these impeller will start hitting the walls of the fermenter. So the impeller should also be smaller than the diameter of the fermenter. But what happens is that this impeller is going to mix the contents only within its reach. It is not going to mix the content that is present at this part or at here or at here. So this process is called as vortex. This phenomena is called vortex. When the impeller is going to mix the contents which are only up to its reach, that is called vortex. And to prevent this vortex, baffles are inoculated, baffles are kept here, so that the contents which are present at this part and at this portion of the fermenter, they will be mixed properly as well. So that is why, to prevent the phenomena of vortex, baffles are used. Okay, so I already discussed these points. Yeah, one more point, that four baffles are used because fermenters are very large. Using one, two, or three baffles will not be enough, so four baffles are used. And as I said, that baffles are the metal stripes. They have to be 1/10 of the vessel diameter, because if they are not 1/10 of the vessel diameter, then the baffles will start colliding with the blades of the fermenter, and both will hinder each other's activity. Okay. It is said that when the baffles are wider, the mixing is increased. But when narrow baffles are used, the activity, the agitation, the mixing done by the baffles will also decrease. So that is why it is advised to fit wider baffles. Okay. And the last part of the fermenter, that is the sparger, or we can say the aeration system. So this is how a sparger looks like. This is a sparger, and this is the diagram showing sparger present inside the fermenter. So spargers are actually, they are metal ring. If we can see clearly that these are the metal rings. Yeah, so these are the metal rings, as we can see, and these metal rings have small small holes in them. As we can see, these are the small small pores here. Small small holes are there. So what is the significance of sparger is that I said oxygen is to be inoculated inside the fermenter. So it is the sparger through which oxygen will be inoculated inside the fermenter at a very high pressure. So these are the holes through which the oxygen is going to get inside the inside the fermenter at a very high pressure. Now, whenever oxygen is inoculated in the fermenter, why high pressure is used? High pressure is used to avoid contamination, and also to mix the contents, because when you are inoculating anything at a high pressure, the media will jump a little bit and it can mix on its own as well. So that is why air is always inoculated, oxygen is always inoculated through the sparger at a very high pressure to avoid contamination. So this is how a sparger looks like, and these are the various holes through which oxygen is inoculated. Okay. So I already said this point, that spargers are used to introduce air inside the fermenter, inside the production media. And as I said, the spargers are a series of holes in the metal ring. So these are the metal ring, and there are, as I said, that these are the holes, or we can say nozzles. And oxygen enriched air passes into the fermenter under high pressure. So I already said this point as well. Okay. So with this, I complete my discussion on fermentation. What is fermentation? What are fermenters? We saw various parameters, steps of fermentation, and how an ideal fermenter looks like. Various parts of the fermenter is also there. So before ending my session, I have prepared some questions as well. These questions, so there is a small quiz. These questions are prepared by me only. So we will solve them properly, and you can answer them by unmuting yourselves or writing in the chat box. Okay. 1 second. Yeah. uh, Is my PPT, is my presentation visible?
Yeah, Richa, yeah.
It's okay. So this small quiz I have prepared. Okay. The first question is that fermenters are also termed as dash. Options are bioreactor, bioprocessor, both A and B, and none of the above. You can either unmute yourselves or you can write it in the chat box as well. Anything can be fine. Okay. Only one answer. Uh, okay. Okay. Giving one more minute for you all to answer it. Then we will move to the answer. Okay. The answer to this question actually. Okay. I have got one more answer here. Okay. Actually, the answer to this question will be the first option, that is bioreactor. For why bioprocessor is not the answer, let me tell that. Bioprocessor is actually the term we use when for bioreactors. The mini sized bioreactors are actually called bioprocessor, but fermenters are not called as bioprocessor. The small size of bioreactors are actually called bioprocessor. So the answer to this question will be option A, that is bioreactor. Okay. Next question is internal surface should be dash inside the fermenter. Options are rough, smooth, both A and B, none of the above. Okay. 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 So moving to the answer. The answer is actually smooth. Internal surfaces should be smooth in the fermented, because in one of the slides I said that when we are constructing the body of the fermenter, we cannot use a single sheet of stainless steel. We have to use two or more than two sheets, and to join those two sheets there are two options. We can either do welding or we can fit screws into them, but fitting screws will create a problem because the production media will start accumulating all around the screws, and also the microorganisms will start gathering on those screws. So we actually prefer internal surfaces, smooth surfaces, and to provide smooth surfaces, welding is the desired ones, because because in welding we are heating and we are melting the metal sheets to join them. This is going to give us a proper complete smooth surface where production media or microorganisms will not be sticking, because what happens by using screws, it will give a bumpy surface at the internal surface of the fermenter. Those bumpy surfaces are actually not desirable. So the answer is actually smooth for this. The next question is impeller is used for dash, agitation or mixing, separation, aeration, both A and C. Okay. 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 The answer. The answer to this question that impeller is used for dash, the answer is actually option A, that is agitation and mixing. Okay, let me tell that why separation or addition is not the answer, because as we saw already that these are the impellers which are actually used for mixing, and the other two terms here are separation and aeration. As I said, that when mixing is taking place, we are supposed to create a homogenized mixture. We are not supposed to separate anything. So to create an homogenized mixture, agitation is important, that is mixing is important. And why aeration cannot be the answer is because for aeration spargers are used. These are the sparger. Impeller cannot be used to provide oxygen. Impellers cannot be used to inoculate oxygen inside the fermenter tanks. It is the sparger through which oxygen is to be put inside. So the answer to this is agitation and mixing. Next, fourth is fermenter is constructed by dash material, glass, stainless steel, both A and B, and none of the above. Okay. Okay. The so moving to the answer. So fermenter is constructed by dash material. So the answer to this question is stainless steel, because fermenters are the large bodies which are used in the industries. If anytime you visit Amul industry, which is in Gujarat here, or Coca-Cola industry, which is also in Gujarat, I have visited these two industries, and what I have seen is all the fermenters are made up of stainless steel only. It is only the lab scale fermenter that is made up of glass. So the answer to this question um, is stainless steel. Okay. The next question is usually dash baffles are inoculated. Options are 1, 2, 3, and 4. Okay. Okay. The answer, uh, 1 minute. Yeah, Om Prakash sir is right. Four baffles are used because yeah, because right now I said that fermenters are very big, they have large diameters, and to cover such a large fermenter, one baffle, two baffles, three baffles will not be sufficient. So it is the answer is four baffles. And the last question is use of flange points, that is, flange points are screws. So use of screws should be maximized. Answer options are true or false. Okay, I have one answer here, true. Okay. Flange points would mean screws. Uh, okay. Okay, moving to the answer. So use of flange points should be maximized. The answer is actually false. The question says that use of flange point should be maximized. That means we have to use screws more and more. And just now I said that if we use screws, it will provide a bumpy surface. The internal surface will not be smooth. Production media will start accumulating around the screws. Even the microorganisms will also start gathering around the screws. So here we will not be able to get sufficient amount of product, because let's suppose that if half of the organisms is sticking in all the screws, then only there are a handful of microorganisms which is present in the actual fermenter. So we will not get sufficient product because most of the microorganisms have sticked around the screws. So that is why flange points use should not be maximized. Welding use of welding is to be maximized, is to be used more. So yeah, so with this, I complete my topic on fermentation equipments and its use. Thank you. If anyone is having any doubts or any difficulties, we can you can start asking if there is any doubts or anything. Okay.
I think Richa, no one of them have any doubt.
Yes.
Uh, when before starting the session, I have just dropped one of the message in the chat box. One of the I have replied to one of the member that certificates are not going to provided by the AgriExplorer to these sessions. Okay, so most of the people have left the session. No problem. These are the learning sessions, and these are for the students. You have presented well, and
Thank you, sir.
I have one of the doubt regarding
Yes,
the
sir.
question number two.
One
Uh,
second, sir. Yes, sir.
According to me, the fermenter should have the rough surface because the rough surface increases the surface area. And if the surface area increases, the bacterial activity or activity of the microorganisms increases. Uh, so I
Yes,
think the answer should be rough. If it is smooth, please explain why.
yes, sir. Actually, when I say that internal surface of the fermenter should be smooth, that means the walls of the fermenter should be smooth. Not the We are not talking here about the base of the fermenter. So, like in this diagram, we are actually talking about the walls of this fermenter. The walls should be smooth. Base can be anything, but these walls have to be smooth. Now, if they are rough, then what will happen is that if the surface is rough, when the process of agitation is taking place, the microorganisms will start sticking at on the rough surface, because microorganisms will get a rough platform to stick. Whereas if the surface is smooth, then microorganisms will not be able to stick on the smooth surface. They will start slipping down because the surfaces are very smooth. They will start slipping. But if it is rough, it will be an ideal situation for the microorganisms to stick on the walls. And if let's suppose that most of the microorganisms start sticking on the rough walls, then product will not be formed properly, because this is the main area where the process has to take place. But if let's suppose that half of the microorganisms start sticking on their rough walls, then the sufficient amount of product that will not be made. So the answer to this will be smooth, because here we are talking about the walls of the fermenter, the inner walls.
Yeah, okay, understood. But I think I have read somewhere. I think it was IIT Roorkee, one of the course I have attended. I think I have heard that rough, but I will confirm it.
Okay, sir.
Okay. So I think anyone don't have any doubt regarding the presentation, but your presentation was excellent. Your
Thank you.
skills of understanding, making the people to understand are good. So I congratulate you from the Agri Explorer, and I hereby declare that uh, the session is